barrier in our brain to protect the cerebral spinal fluid system into the bloodstream. This blood-brain barrier is very difficult to get substances across if they are polarized or a large molecule. So proteins or ions have a difficult time traveling across the blood-brain barrier. When we look at the cerebral spinal fluid, it's made by ependymal cells. These ependymal cells are highly concentrated in the choroid plexi of the ventricles. And ventricle and the arachnoid space of the brain. We then started talking about the brainstem. And when we were thinking of the brainstem, the part closest to the spinal cord is the oblongata. The next closest part to the superior is going to be the pons. And this is where we left off last Friday with the pons and the midbrain. As we look at the midbrain, some key structures on the midbrain include the corpora quadrigemini. These corpora quadrigemini are going to be involved with regulating some motor movements. We also are going to have a lot of motor nuclei. To reiterate, when we talk about nuclei in the brain, a nuclei is a region of gray matter surrounded by white matter within the central nervous system. The analogous structure in the peripheral nervous system is a ganglia. We have the dorsal root ganglia, or the posterior root ganglia, to use the new school term, within the spinal nerves. But a grouping of cell bodies of neurons in the central nervous system we refer to as a nuclei. From the midbrain, there are two key cranial nerves that control eye movement, the ocular motor and the trochlear nerves that originate there. And this is new material. We did not cover this slide last Friday. So as we look at our midbrain, at the midbrain there are 4 bumps. The posterior of the midbrain, referred to as the corpora quadrigemini. These bumps are the colliculi. We have the superior and inferior colliculi. On our diagram, our corpora quadrigemini are going to be right here. And they are the most superior part of the midbrain. There's a small mid-sagittally located bump directly above that. That's going to be the pineal gland. And our pineal gland, which is right there, directly above the corpora quadrigemini, that's categorized as part of the diencephalon. Now, when we look at these corpora quadrigemini, they're going to allow for us to have reflex tracking. They allow for us to monitor and regulate motor movements. And they're also going to allow us to relay information from the inner ear. When we think of inner ear, I want you to think of equilibrium. We'll talk more about that on Wednesday and Monday and Wednesday of next week, our last 2 lectures next week. We also have, on the anterior portion of the midbrain, some tracts, and these tracts are going to extend superiorly. These fibers or tracts are referred to as the cerebral peduncles. And if we look at the root words of the cerebral peduncles, they are the pedestal that the cerebrum or the brain sits on. They look kind of like a column that holds up the brain. The cerebral peduncles are going to have the tegentum, substantia nigra, and the cerebral crux as part of them. And as we look at our midbrain from this viewpoint, we have the corpora quadrigemini, visible right here from the mid-sagittal view. Here's the pineal gland of the diencephalon. And then the cerebral peduncles are going to make up the primary mass of our midbrain. When we look at the cerebral peduncles, let's talk about them in a little bit more detail, the functions of the cerebral peduncles. We have the tegentum, and when we think of the tegentum of the cerebral peduncles, most individuals use tegentum and red nuclei as synonyms. The red nucleus is a region of neurosomas within the peduncles that's highly vascularized neurosomas, hence the red color. There's a lot of blood supply there. And when we look at this area, we regulate and modulate motor control. So in green, I'm circulating the red nucleus, which is effectively also known as the tegentum. We also have the substantia nigra. If we look at the root word here for niger, it means black. The substantia nigra is a region that is pigmented black. This region that has dark pigmentation is pigmented darkly because of the high concentration of melanin, the pigment melanin, not melatonin, the hormone. And when we look at the substantia nigra, they help to regulate and reduce the amount of tremors that occur in our bodies. The substantia nigra is going to interface with two other structures we haven't talked about yet, the thalamus and the basal nuclei. These substantia nigra will work together with the thalamus and the basal nuclei to reduce the amount of tremors that occur. We refer to these typically as intention tremors. And when we think of the degradation of either the basal nuclei or the substantia nigra, we're typically going to have a lot of unregulated, jerky motor movements associated with the brain. The classic symptom of this is going to be Parkinson's disease. So when we look at the beta amyloid plaque buildup in the brain associated with Parkinson's disease, these beta amyloid plaques will preferentially destroy neurons within the substantia nigra, thalamus, and the basal nuclei. We also are going to come back to that reticular formation. The reticular formation is a loose spider web or network of gray matter that runs all the way up through the brainstem, through the diencephalon, all the way to the cerebrum. And when we look at this reticular formation, it allows for information processing to occur as we send information to the thalamus, and then from the thalamus, that information needs to be sent up to the cerebrum for the final decision-making processes to occur. Now, when we look at this network of fibers, this reticular formation, it helps, the processing isn't necessarily going to be the decision-making. Most decision-making, per se, is going to happen in the cerebral cortex itself. So within the superficial portion of the cerebrum, that's where most of the decision-making happens. The processing that occurs with this gray matter is going to be, well, where should I send the signal? Should I send it to the occipital lobe to further process? Should I send the signal to the parietal lobe? Should I send it to the frontal lobe? So that's very low-level processing that occurs within the reticular formation. The processing determines where to send the signal next. Some of the functions of a reticular network include regulating motor control. When we think of somatic motor control, I want you to think of both autonomic and voluntary. So some of this is going to just happen automatically. Some of it we can have voluntary control over. A key component of this is muscle tension. We have low-level contractions throughout our entire bodies right now with our skeletal muscle tissue, and these low-level contractions are going to be regulated by the reticular formation. We're also going to have signals from our eyes and ears to help us with our sense of balance. You probably noticed within your inner ear you have an equilibrium apparatus. And if you spend any amount of time spinning in circles, you get dizzy, car sick. Your equilibrium apparatus in your inner ear senses that you're stationary, but your eyes are perceiving that you're moving. And when your eyes and ears are working together, you typically feel fine. But when you have that disconnect between the information, the reticular formation has a hard time processing it, and a lot of people will feel car sick or motion sick as a result. We're also going to use the reticular formation to help regulate gaze centers. For our eyes to fixate on something and move and have both eyes fixate and track an object, that takes a lot of automatic control. We don't think about the 6 muscles associated with each eyeball, just that we use 12 muscles working in a coordinated fashion, and most of that just happens automatically. We're also going to have some central pattern generators. I want you to think of movements that happen automatically. The contraction of your diaphragm, breathing in, breathing out. Movements or muscle contractions that happen rhythmically throughout your lifespan, throughout the day, regulated by those central pattern generators. We also use, in addition to this regulate cardiovascular system. So we're going to use them to regulate the heart rate. We're also going to use them to regulate contraction of the tunica media, the muscle of the blood vessels. So we're going to control blood pressure through the reticular formation by changing the amount of blood that we pump out of our heart and by changing how tightly our blood vessels squeeze the blood. If we constrict the smooth muscle of our blood, then we have higher blood pressure. If we relax it, we have lower blood pressure. And then one of the more interesting pathways for me in particular is pain modulation. You probably, if you've spent any amount of time in a high stress or high anxiety situation, not feel the pain. And many student athletes have experienced this before, where they get an injury during the competition and they don't even notice it. They just keep going. The next day, you notice it, and it hurts a lot the next day. One of the reasons for that is because we have the ability to turn off sensory signals. We can selectively turn off our ability to perceive pain. And part of this ability is going to be regulated with the reticular formation. So we are going to have the ability to modulate the signals as they're ascending, as they're going up to the cerebrum, and we also have the ability to send inhibitory signals to the sensory receptors. Additional functions of the reticular formation include regulating sleep and consciousness. The difference between being asleep and awake, it's a very fine line. And this reticular formation serves to integrate all parts of the brain loosely and will help to regulate our ability to fall asleep, our ability to be alert, or maybe our ability to be awake but not necessarily alert. If individuals have damage to their reticular formation, sometimes that results in them having a coma and not being able to wake up. The reasons for this are not very well understood. One of the big mysteries right now, one of the million-dollar questions is, why do we have to sleep? And we know that sleeping's good for us. We know that if you don't sleep, bad things happen. But why on earth do we have to sleep? By all accounts, there's the appearance of not needing to sleep. We have all the mechanisms in play to circulate and maintain our tissues and cells when we're awake, so why can't we just be awake all the time? And these are some of the mysteries that we have right now with our nervous system. And then finally, we also have habituation. And when we think of habituation, I want you to think of filtering out repetitive stimuli. Habituation is very closely related to this idea of breaking in your shoes. You've probably gone through the experience of buying a brand new pair of shoes. And you put on that brand new pair of shoes, and it kind of hurts. It doesn't feel quite right. You put it on, you maybe for a couple runs, you wear it for a couple of weeks, and after a couple of weeks, that new pair of shoes feels good and comfortable. This process of breaking in the new pair of shoes is really breaking in your brain. You need to habituate to how it feels so that you can focus on the important things. Typically, when we break in shoes, we're not changing the shape of the shoe so much as we're changing our perception of how the shoe feels on our feet. So, this review question was meant to be covered on Friday last week. Anyways, which structures create the blood-brain barrier? You'll want to flip back to the earlier part of this presentation. So we're down to about 30 seconds. We have gap junctions between endothelial cells, tight junctions between endothelial cells, gap and tight junctions between astrocytes and endothelial cells, desmosomes, or gap junctions between endothelial and ependymal cells. Which structure creates the blood-brain barrier? We're at about T-minus 10. Make sure to get your answers in so you can receive your participation credit. Guys, gals, as we think of the blood-brain barrier, I want you to think of astrocytes stimulating the endothelial cells to make tight junctions. So we have endothelial cells forming tight junctions or waterproof junctions. The astrocytes are important. They stimulate the generation of those tight junctions by the endothelial cells. And it looks like this is a little bit harder of a question. That is all for the overview of the brain and the brain stem. Let's move on to the next segment. We're going to focus on the cerebrum and cerebellum, the next parts of our brain.